the plant decellularization for tissue engineering scaffolds. My professor is Kaigo Kawaji. He works at IIT and he does several projects like MRI system. He made it more efficient to use in clinical situations. He also focuses on mathematical problem solving ideas and techniques, which he tried implementing on us through puzzles. A specific paper he wrote recently is detecting motion in real-time cardiovascular MRIs. But today we're going to focus on tissue regeneration, which he has done work on too. The paper we're focusing on and following is Crossing Kingdoms using decellularized plants as perfusible tissue engineering scaffolds. And you can search this up, you can read it for free online, but I can give a background here. So scientists have used bioengineered scaffolds or just structures for tissue engineering to help out with patients for skin grafts or similar needs. And they used animal tissues or just human tissues, which have been rare to find and more expensive. So the professors in this paper used similarities in vascular structure of plants to see if they could replicate what they did with animal tissues before. And different plants have different geometries, so they'll have different structures that can fill out the human skin cells that they use after. And as I said before, this is more cost efficient and also more green technology. And as they wrote this paper, it worked out. So this is kind of the process how they did it. They used a syringe to cannulate the leaf here, and then they bleached it so they can recellularize with human cells after. My project is similar in that I used spinach, kale, and parsley, which are like different plants, to measure different geometries. But I used shampoo with sodium lauryl sulfate instead of the actual chemical itself, because like shampoo is more easier to find. And I used the same bleach and I soaked it in the 10% solution, similar to they did in the paper, except they cannulated it instead of soaking it like I did. And they also switched out the solution every few days, but I mixed the solution every few hours to avoid the heterogeneous mixture. And I kept a lab notebook throughout this, as he asked us to do, and this has all the same parts as you would have in a normal science experiment, like the hypothesis, materials, procedure, constants, to ensure that I could reproduce this experiment after. So what decellularization actually is the process of stripping off cells of genetic material from tissue. And you can see that through this liver diagram here, where the cells are removed, leaving behind the extracellular material, which is the structural scaffolding. So these are my observations that I did myself. Day one is these plants without the prior to the solutions, and day two you can already see how some of the cellular material is dissolving into the solution, and you can see kale is the most green solution while parsley is lagging behind, which is consistent throughout the experiment. This is day three. You can see all of the plants have been almost transparent, and parsley is more delicate than the others. This is what we're looking for in a decellularized experiment. This is day 4 after I started bleaching. You can already see how the day, like in the evening after, you can see spinach is turning whiter, like losing its chlorophyll. Same with parts of kale, but not the same with parsley. Day 7, you can see kale and spinach being almost completely white, except for the stems, and interestingly parsley has whiter stems with almost no white in the leaves. And this is day 19, marking like the end of the experiment. This is like currently, you can see all the plants have been turned white and have lost all the cellular material. So what are the implications of this experiment? Uh, Tissue scaffolds and the structures we made can be used as vascular grafts and can be used for surgeries and regenerative medicine. And we tested out different plants which showed different geometries, and each plant has different efficiencies and durability. For example, you saw how kale and spinach were more efficient than parsley, and parsley was less durable too. So that could show how parsley is maybe not the right fit for such a vascular graft. And some of the next batches we're going to do, we might test different ways, like procedures to do this experiment, or different other plants or vegetables. And when we actually get to IIT, we can assess how much of our scaffolds have been decellularized and how much of it actually works. And that's it. Thank you.